Yeah, I just want to, I'm the part of the primer that is really just here to give you an idea of what the sequencing technology is about and what we really want to do with it overall. Warren's really the meat of this. I'm just trying to demonstrate how some of this could be useful to you in a non-condescending manner. So, and we'll see how it goes. Right. This is just an illustration from genome.gov of a chromosome unwound into a long DNA molecule, and really the idea of what we're trying to get at overall in terms of genome sequencing. We're trying to get at a read of these base pairs and their complements on another strand, that we can understand basically what all genetic code we have along that whole chromosome. And so the real idea is to take that and read every chromosome, every bovine chromosome in this case, and understand what that chromosome reads from one end to the other in terms of the number of bases. Okay? Whole genome sequencing then really is loosely defined as reading and recording all 3 billion of these bases in the cattle genome. And I'm kind of skipping over a lot of details here, but generally the genome's made up of 2 different types that I'm going to categorize right here. 2 of them aren't exactly, one of them's not exactly the same thing, but part of the genome, over 98% of it, are regions that actually don't code for any protein specifically. Okay, that's either intronic regions, non-coding regions that are within the genes that make the proteins, or intergenic regions. And we don't know what a lot of that is necessarily. I'm not going to say it's not useful at all, but it does not code for the protein itself. Likely there are some regulatory, we know about several regulatory functions and things in these regions, but it doesn't actually code for the proteins that we're interested in, at least in the short term. Exons are the remainder, approximately 2%, and these are the coding regions that bring about the proteins that are used in the building blocks of the mechanics of our cells and of life. And this is sort of a quick demonstration. I don't want to spend too much time on this, but of that process of basically we get a transcription from DNA to an mRNA molecule, which is basically complementary bases of that DNA that we started out with, and through the intracellular mechanisms in the ribosomes, we end up with an amino acid chain that, when sometimes by itself, sometimes combined with other chains, becomes a protein at some point. And these are the proteins then that eventually function as enzymes to help chemical reactions and the physiological mechanisms inside our body go on, the building blocks for several of our muscles, things like that. And basically, the important thing here is, as we read along here, we see that these amino acids bind to what's called a tRNA molecule that is coded in 3 base reads. And so every 3 base set that runs through that ribosome is really coding for a complementary molecule that brings along a specific amino acid. There is some redundancy here. There are several of these 3 base bases groups, 3 base groups called a codon, that actually code for multiple, or called for the same amino acid. But the point is really that there is some specificity to these 3 bases, every 3 bases, and that builds something in our chain. And the variation then that we see in a lot of our phenotypic characteristics really comes about because that chain is being built differently based on what this base pair is coding for in that chain. Okay, that's the point I want to get across here, is that what we see as differences oftentimes are due to how that chain is built. And it's part of what we're interested in the short term then with sequencing, and what we hope to get a little bit further with in several projects that are going on right now. This is another summary of that, and a summary of what happens with a mutation, and basically what can happen then in terms of genomic variation when one of those bases switches out. Here we have several 3 base codons that are all coding for the same amino acid here, okay? And you'll see that, just in this example, if there is a mutation at some point where one of these codons changes one of the bases, we end up with a different amino acid, which may cause or may not cause a malfunction in that protein. May cause the efficacy of that protein to change, may cause nothing at all in some cases, and the protein functions the same way, may cause the animal to die in some cases because the protein needs to work correctly in order for the animal to survive, okay? This one's actually relatively non-severe in general. One base change and an amino acid change is a little more common. Sometimes changing a base does not cause a change in the amino acid change at all because of the redundancy I mentioned in coding for amino acids, okay? This is an example of a mutation then called a frameshift mutation that's a lot more severe. And in this case, instead, we really have a frame reading difference where instead of reading CAT, TCA, the way this chain is being read across, probably because of a deletion somewhere upstream, is being read as ATT, CAC, and as you can see, all of these amino acids are completely different along here. This is a much more severe mutation. Basically, we get a completely different building block of a protein being made here. Would cause very different effects if this protein is important for some sort of physiological process, which is generally the case, and is probably responsible then for a lot of the variation that we see. And there are several things like this that can happen that we'd call severe variants, where we have a big change like this, where we might end up with something called a stop codon, where instead of reading and continuing to extend the change, perhaps something right here changes and the amino acid change stops. There's a signal, a 3-base codon signal that would tell it to stop making the protein. And so these are the kinds of things as we start to go into our sequencing technologies that we want to look at and use as tools to see if we can discover a little bit more what's going on underneath that might be causing the changes in the phenotypes that we see in our cattle species today, okay? History a little bit on genome sequence. The first animal was a line 1 Hereford from Miles City, Montana, named Dominette. Pretty inbred animal, I believe. I can't remember exactly what the inbreeding coefficient was calculated, but they're easily up to 40% in some of the line 1 cattle up there. This cost about $53 million to get done at the time. It's been served as the reference bovine genome in 2 different builds primarily today, and it's led to really what we get in all our Illumina chip technology that we see today, okay? It's the base also then for further sequencing projects right now. And this, I don't want to spend much time on this again, but this just kind of shows where some of those costs are coming from in some of the technology, and I'm not going to spend much time on this other than to show you as we progress through time here, even at the US Meat Research Center, what was happening to the cost per million bases being read as we progress through technologies over time. And you'll see that we went to, somewhere just around 8% or so, sorry, more than that, 20% or so of the cost from 1992 to 2003. And what's happened since then is really technologies called next generation sequencing that have cheapened the cost quite a bit and result in high throughput output of the genome sequence and made this whole ordeal a whole lot cheaper overall. In fact, we've ended up with costs that have gone from what I was mentioning per million bases in the thousands to somewhere around between 5 cents and $10 per megabase now, okay? So we're talking about several orders of magnitude different in the cost of doing that. It really is why we're talking about sequencing animals today in the scale that we're talking about. You'll see on this slide as well, accuracy can be a problem depending on the platform going on here. I don't want to get deep into that necessarily for you today other than to say generally that's a function of the read length. not strictly speaking, but generally a function of the read length. As the longer the reads are of sequence of bases being read, the less the accuracy can sometimes decrease. There are ways we can account for that. I know that 87% doesn't seem very high, but due to multiple readings and different types of technology, we can account for those accuracy and those errors. And basically, it helps us to move forward with this technology overall. Okay. There are some complications. The sequencer obviously costs a lot of money to get started with this, from $80,000 to $1 million. So we still do sink quite a bit into this, but a lot of that's being taken care of because one group buys a sequencer and then they contract out these sorts of things and make the technology cheaper for the rest of us to do that. The one I want to focus on just a little bit here is the Genome Assembly. And this is just a small graphical demonstration of that from Roslin Institute slide, where if we take the chromosomes that we start out with, and you'll see by the colors sort of what those look like and how they should be arranged if we were able to put this jigsaw puzzle back together, break it down into the small read lengths and fragments that they're actually being read into, we've got to figure out a way to get from here down to something that we can put back together again and figure out how those bases actually read across. And what you'll notice about this, and this is important, is that here we've got 5 different sorts of lines that we're trying to build out of this by the way these pieces are broken up. And if you look at different parts, for instance right here, we may only have one sort of read that actually covers that target. Okay, so these are some of the complications in trying to put this sort of thing back together for the whole genome out of tiny little pieces and trying to figure out where those pieces should go and what actually the read would be for that animal when we only read it once. Okay. Overlapping helps. Longer reads are better. Where I mentioned that we lose accuracy with longer reads, what they do help us do is give us a lot of overlap between the reads so that we can see based on the base pairs that are at the end of one segment and the base pairs that are at the beginning of another segment, how they should fit together a little bit better. It also helps to compare to a base sequence of other cattle that have always been sequenced, and that's where that initial project was so important with the female dominant. Okay. There's a tremendous amount of data to worry about. It's hard to store, hard to find enough storage dates to do this overall, let alone manipulate the data and try to put everything together, which makes this assembly process even tougher. And then we have to develop tools based on the results. For instance, we need to discover ways to genotype based on new markers that are discovered in the sequence. That's what part of the 50K was based on originally, although there's other methods that went into that as well. It's hard to decide what differences among animals are important, and as with chips, if we're going to use this sequence data well, we need to continue developing phenotypic resource populations so that we can actually take advantage of the differences we see in the sequence. Other complications, there's multiple reads required. Just because we read 3 billion bases does not mean the whole genome is going to be read. This is also a function of the random process of those small little bits of reads that I was showing you a little bit before. Just because we read 3 billion bases means we're generally reading several of those over again. And I think there was some math thrown out a little earlier, maybe that approximately a third of the genome would be read once if we read 3 billion bases one time. Approximately a third would be read multiple times, and approximately a third might not be read at all. Okay, and that's just sort of general math, but it shows some of the complications, and so that's why sometimes we get advocates if we want to sequence one individual animal of sequencing up to 20 or even 50x when we target the whole genome. Focusing on one animal, though, reduces the impact of examining multiple animals because of the cost of sequencing in the first place. Okay, so there's a balance that we all need to try to strike here on the best way to go forward. We're really trying to feel out, do we want more animals with less coverage, or do we want fewer animals that we really know more about? Okay. From a geneticist perspective, why are we really interested and what are we bothering? Why are we bothering with this? We're interested in sequencing to improve our chance at finding causal variation. That's really what this is all about. We've had these chip technologies in our hand now for several years. They've done a tremendous service to the industry overall. Mostly they're just markers. And it's part of why we don't get them to work across breeds. It's partly why we have such a difficult time in future generations, thinking that we may have to retrain and recalibrate all the time. And what we really would like to do is get to things that are a little better and a little more causative, and understand how we can go forward so that it doesn't change and we don't have to retrain quite as much, okay? One way to do that, and what might be the low-hanging fruit now, is what I mentioned before then, trying to look at mutations that actually cause changes in the protein structure or protein regulation. And ultimately then the differences that we see are due to the genetics that lie within the differences of the genome, okay? There's been two different strategies going about sequencing. I'd say one is a little older strategy and one's a little newer, and this is from my perspective. I'm sure other people could think of other strategies. But one has been to compare genomes of animals that are diverse in their phenotypes and look for differences in them to decide based on how different they are in their genome sequence, what mutational candidates we might have, and then for polymorphisms that actually cause the differences that we see. So for instance, take high feed efficiency animals, low feed efficiency animals, look at their differences in sequence, and where we have differences in the sequence, those are candidates for future genotyping. They may not be real, they may be by chance, but they're a way to get started. The other way, and this is the way I think we're starting to go a little bit more recently, is look at differences in the sequence among several animals and try to associate that with trace variation. And this gets back to maybe looking at some of those protein encoding regions and trying to get at a little bit more about those variants that are very severe, and associating that with the phenotypic databases that we have large amounts of, okay? These are some examples, and I'll move into Warren's part here. This is just some examples of some of the variants he talked about. I don't want to go deep into what each of these things are, other than to kind of say how similar it is to some of that example I showed before, where we have variants with high impact are due to like that frame shift where the whole amino acid changed, or where a start codon is added, or a stop codon is added or lost. An exon is deleted completely, things that really mess up a protein, okay? And then moderately, these might be some of like that first mutation I saw where one amino acid changes in the whole chain, okay? And it can go down from there. So with that, we'll move on to Warren's part and where we're going right now at the Meat Animal Research Center in our sequencing efforts. I do want to mention there's other important sequencing efforts going on. Jerry's got a good project going on in reproduction right now. I know Dorian at Iowa State's working with some of this, and this is kind of what we're doing here right now. So.
All right, thanks, Larry. I think that was a really nice introduction to sequencing in general, and as Larry said, Dr. Warren Snelling will visit a little bit about the activities going on here at the U.S. Meat Animal Research Center. And I think, Larry, as you pointed out, it's interesting that perhaps a bit of a change in the paradigm, starting off with phenotypes and asking the question, how much variation can we describe by these SNPs, versus looking at sequence information saying, hey, we've got a severe variant, what traits it impacting? So, Warren, I'll go ahead and turn it over to you.
Thanks. What we decided here, with our sequencing effort was to, try and get a lot of the breeds that we're using in the germplasm evaluation that is based on the, usage in the U.S., get all of those breeds represented by some sequence, and rather than trying to sequence a few bulls deeply to discover everything about them, we'll try and use this low coverage strategy to detect variation in that exists in this population. Right, what we have right now is data from 167 purebred bulls. Most of them are in the top 7 breeds, what we call cycle 7, with 16 red Angus bulls and 19 to 20 for the more popular breeds. And then the smaller breeds that we just introduced in our new continuous GPE project, we have one bring us up to 7 Solaire bulls. And these were all selected based on number of influence in our population. We also have some F1s bulls that are, most of them are crosses, F1 crosses of the cycle 7 breeds. We do have a few Brahman Angus F1s that were also sequenced. And out of these, 230 some bulls that we've done so far, we've got about 35 total variants that we've detected. 30 million of those are SNP, and they're about 1 million and a half insertions, deletions, and and a few other types of variants. Some it's a single base that you may see three of the 4 possible bases at that location, and then there are some that are more complex than that, involving several bases. And Larry just showed how we categorize these variants relative to the current bovine annotation. There are 95,000 of these that I'll call interesting. They're either those high impact or higher moderate impact. The moderate impact are mostly non-synonymous SNP that cause a change in the amino acid of a protein. So out of that 34 million variants that we've detected, I think we're focusing right now on that 96,000. A little bit more about how well we covered the genome, and from the full data set, we're seeing almost all of the 780,000 SNP that are on the bovine HD chip. And the coverage of those positions is, on average, we covered about each position a little less than once per bull, or a little less than twice per bull. So that means we're not able to call genotypes from sequence on all of these bulls. Where we do have several reads covering a position, we can do a decent job calling genotypes. On average, 4 to 5 reads to have a decent probability of calling both alleles. And if we don't get that coverage, we can call one allele. So this looks at, we're calling about 30% of the high density SNP, from sequence, by bull, or most of the markers are called right around that 30%. There's not many that we call in more than half or there's none that we call in more than half the bulls. There are a few bulls that are well covered. One bull we can call 97% of the high density genotypes from his sequence. There's some bulls that we call less than 10%, and half the bulls we call less than 20% from sequence. Where where we can make calls from sequence, we do get good agreement between the genotype we get from sequence and what's our genotype from the high density. Where we're missing most is on the heterozygous calls, where the reads we have covering a position may all be for one allele. So if we had more coverage of that allele, we would increase chances of seeing the other allele and making the correct homozygous call. And our call rates on these interesting variants that we see in sequence are a little bit lower. 20% relative to the 30% we had with the high density SNP positions. And just thought I'd take a look at alternate allele frequencies where the reference allele is what's in the Hereford's reference sequence, and the alternate is whatever else we saw from the bulls. So the Hereford has lower frequency than the other breeds for both the interesting and high impact variants. But none of these are extremely rare. If you look at the Taurus versus the Indicus cross and Ross Indicus cross bulls that we have in this set, they have higher alternate allele frequencies than the Taurus bulls. So, using the genotypes that we could call from these bulls with their high density genotypes plus high density on another 720 animals in the pedigree, and 8,500 50K genotypes that we have on the rest of the animals. We went through a process like Elisa described to impute the interesting variant genotypes to the rest of the population. So we, with 18,000 animals in the pedigree, we managed to get those genotypes imputed to almost 10,000 animals that had other genotypes. And we do have another source of sequence data that's exome sequence, that a portion of these animals, there are 47 animals in the 10,000 that we have exome sequence for and imputed genotypes. And where we could call genotypes from exome and make a comparison to the imputed genotype. We get 83% of those calls agree, and in the exome, we may be missing. We only see one allele in that sequence, and 85% of the calls are at least consistent with what we imputed from the bull sequence. There are 61 of these bulls that also had exome sequence, and we could do the similar comparison just between their genomic and exome sequence, and we see the similar rate of matching genotype calls, and a fairly high the calls from both data sources are pretty consistent, where we only see one allele. But this shows that if we had increased coverage either through exome or genome, we could make more accurate calls on what an individual's genotype actually is. So one thing we haven't done yet is tried to merge the two sets of sequence to where that would give us more depth, at least covering the coding regions, that should improve our call rates and accuracy of the genotypes we're imputing. We also have some projects doing transcriptome sequence that's reading the sequence of the RNA, and that gives us both information about gene expression and what variants are actually expressed. And we can use this data to do things like look for missing homozygous genotypes where we have high frequency alleles that never show up as homozygotes in an individual. Some examples, we can, using the imputed genotypes, we can associate what the imputed genotype with performance. Tried this for birth weight using training with our cycle 7 animals and predicting the new GPE that includes more breeds in a different set of sires. Picking the top 200 of 4,000 imputed high impact, we get a correlation between the MBV and birth weight and new GPE of 0.74. For comparison, did the same process using the same number of randomly selected SNP and get a lower correlation. Similar sort of thing with the full set of interesting variants and the set of randomly selected SNP. We've also using one set of RNA-seq expression data taken from 16 steers that were high and low in four in each group of high and low intake and efficiency. So, analyses of that data gives 9 genes that were differentially expressed between high and low gain, 15 between high and low intake, and 6 genes between high and low efficiency. And within those genes, we found 70 of our interesting variants that were seen both in the bulls and these 16 steers, smaller numbers for the intake and efficiency. We also see a different set of variants that were seen in the expression data and also the bull sequence. That a lot of these are not necessarily what we called interesting, that may point out some of the limitations of the current annotation, that we're missing information, about those 30 million that right now we're ignoring. So all of this is with incomplete data and preliminary analysis. But when I look at it, it looks like this project's going to provide us a lot of information that we can use to better explain how variation in the genome does affect performance. The sequences we have collected represent the predominant breeds in the U.S., and we can impute the variation we detect in bulls to their progeny, and with enough data we can probably at least do within breed imputation to breeds to individuals that aren't close relatives of whatever we have sequenced. And this data is going to supplement our phenotypic information, what we can get from expression data, and be an important resource to uncover variation affecting important traits. So that's what I have.